Hi, everybody. I'm Jan Witkowski, here a professor at Cold Spring Harbor Laboratory. This is the occasion of the 85th Cold Spring Harbor Symposium on Quantitative Biology. The topic this year is biological timekeeping. It's actually a follow-up to a meeting that was held in 2007, and one even before then, way back in 1960, and things have certainly changed since then. So I have with me this afternoon Stacey Harmer from UC Davis, who is no stranger to Cold Spring Harbor. We were just saying a moment ago that she was here for the 2007 symposium, which was on plant biology. And Stacey, you were saying you were here even before that.
Yeah, so my first visit to Cold Spring Harbor was back in 1998 when I was a student in the plant biology course. And that was a great experience for me because I was transitioning. I had done my PhD in an animal system and I was transitioning to plants for my postdoc. And so taking that course was a really great introduction, sort of a boot camp for getting into plant biology.
Yeah, I think that's actually quite a common feature of those courses, that people take them to learn about a completely new field and change direction.
Right. Yeah, I know it was such a stellar experience. It was an amazing group of students, and we had just a wonderful cast of instructors as well. It was really just a great introduction to the field. Did
you begin working on circadian rhythms and so on immediately after that? Was that have you worked on circadian rhythms for sort of the whole of your professional career?
No, I did some in my PhD. I worked on immune signaling in mouse cells. And so when I made my postdoc, it was a transition both organismally and also topically. So I started working on clocks at the same time I started working on plants.
So you have been really doing it for most of your professional career.
That's right. That's right. And I would add, actually, one of the things I think is really special about circadian biology is it is so explicitly integrative across fields. I mean, across what are often barriers in terms of what organism you study. I don't know if you had a chance to attend much of the meeting, but we had talks just running the biological spectrum from cyanobacteria to mice.
Well, let me, there's a quote I could read from the summary of the 2007 symposium that Michael Menaker did, which sort of relates to what you've just said. He asked in his very last sentence of the summary, how do animals, plants, fungi, and bacteria make adaptive use of their biological clocks in the world in which they live? Which I think is a wonderful sort of statement about what this field is all about.
Right, absolutely. And so I mentioned organisms, but also the talks ran the spectrum from structural biology and biophysics to almost ecological type studies.
And you're particularly concerned about the mechanisms controlling these sorts of biological rhythms in plants.
Yeah, absolutely. Focused on plants, and I got into the field with a very molecular focus, and that's what my talk this year was on. But I've also become very interested in, as you said in that quote from Mike Menaker, what the point of the clock is, what it does for a plant. And so we've also done studies in the field, and in fact looked at interactions between plants and their pollinators. So I'm starting to be a little bit broader in my outlook as well.
And even within plants, you've worked on two really very different, at least in size, Arabidopsis, which is 2 or 3 inches high, and sunflowers that could be feet high.
Yeah. So I started out with Arabidopsis, and I think it was an appealing at the time. It was appealing to me because back, in 1998, the genome was being sequenced for Arabidopsis, so it was kind of the dawn of the genomics age.
Mhm.
So it was clear it was going to be a very powerful molecular and genetic system. And that was a good kind of transition for someone like myself that had been working with cell culture lines, doing biochemistry and molecular biology. And then now that I've worked with plants for many years, it's just my interest have broadened a little bit, that now I'm wanting to look at how they are, how they function growing in a natural environment. And sunflower in many ways is a more tractable plant for those kinds of studies than Arabidopsis.
But can you do this? Does sunflower have the same sort of genetics as Arabidopsis?
Not at
Or
all.
does that not matter now? One has CRISPR and so on.
It's frustrating in a way, because yes, you put your finger on it. Sunflower, we don't have these amazing mutant collections. We don't have hundreds of labs generating a variety of tools. Yeah, and in fact, transformation in sunflower is quite difficult, so that even makes it hard to generate CRISPR mutants. So that's all quite frustrating. But the biology is fascinating. But sunflower does a lot of things that Arabidopsis does not do. And also in some ways, the size of the plants makes some of the experiments easier than trying to do the same thing with Arabidopsis.
Well, maybe we should come back to the sunflower and pollination at the end. Can you tell us a little bit about what your presentation at the symposium was?
Yeah, so I guess I can start. When I was a postdoc, and many years ago now, I identified an Arabidopsis mutant where the clock ran faster than wild type. And when we identified the gene responsible for that phenotype, we found it was a protein that's highly conserved across eukaryotes. So all the way from yeast, from fission yeast, up through mammals. But the protein had no known function, and the sequence of it didn't particularly give us any clues as to what it was doing. So it didn't have any recognizable functional domains. And so we've been kind of chipping away on the problem for the past 15 years, trying to understand what the protein does, like what pathways it functions in, and whether those functions are conserved or not.
So this is a protein that you found relating to circadian rhythms in Arabidopsis.
Correct.
That is conserved across a wide variety of organisms. Mm-hmm. But that immediately sort of begs the question: does this protein fulfill a similar function in these other organisms?
Yeah, exactly. So we've been looking at that, and we found that if we make yeast mutant for this gene, they have a growth phenotype, they divide more slowly. And if we put the plant gene into the yeast, that rescues the growth phenotype. So they clearly have at least some conserved functions. And we've also, in collaboration with the lab of Joanna Chu, also at UC Davis, they generated flies that were deficient for this gene. And those flies do have a circadian clock phenotype, although it's a long period phenotype, not a short period phenotype like we see in plants. So that also suggests that they may have a similar function. And I'll kind of skip ahead a little bit. We found that plants mutant for the gene have splicing defects, splicing of their messenger RNAs. And Joanna's lab has found at least one clock gene in flies is not totally spliced appropriately when they knock down the homolog in flies.
And does this protein affect splicing all cell types, or in Drosophila, or is it very much restricted to the to a subset of genes?
Yes. So they have not taken a single cell approach to that. What they did in their experiment is they knocked down its expression in neurons in general in the head, and then they extracted RNA from fly heads. So, so I can't tell you if it's cell type specific in flies or not.
And the name of this protein, or this?
It's XCT.
It stands for.
XCT.
It stands for.
It stands for ZAP5 circadian timekeeper, and ZAP in this case is XAP as ZAP5, and we named it like it's you have to keep stepping back and back to get to the derivation. We named it after the human homolog that had been identified just as an open reading frame on the X chromosome. So that stood for X chromosome associated protein 5. So we went off of the then name for the human gene.
So tell me a little bit more about how XCT is in, and its role in splicing is carries over into circadian rhythms or biological timekeeping. What's the sort of pathway?
Yeah, well, I have to say we're still not entirely certain. We want to, as a step, to try to understand the function of the protein. A postdoc in the lab, Rod Kumamoto, epitope tagged the Arabidopsis protein, expressed it in Arabidopsis under its native promoter, and then purified the protein and any associated proteins. And then we did mass spec to identify proteins pulled down with XCT. And this mass spec approach allowed us to identify many proteins that together form a complex called the PRP19 complex. And that complex is also highly conserved across eukaryotes. And it's been studied fairly well over the past couple decades. And it turns out that those previous publications told us that that complex, PRP19 complex, has roles in RNA splicing, but it also, components of that complex also have roles in transcriptional initiation and DNA damage repair pathways. So and those things you might think, well, okay, it's because you have a splicing phenotype, every pathway in the cell is perturbed because of the problem in splicing. And that would be a reasonable thing to assume. But actually, those other groups have shown, in some cases, direct, in vitro, with purified proteins, that PRP19, for example, has effects on DNA damage repair pathways completely separate from the effects on RNA processing. So it then becomes, it helps a little bit, like your universe of possibilities is decreased, but you still have multiple ways in which it could be impinging on the circadian system.
So if one looks at this sort of in evolutionary terms, this XCT has it been co-opted for circadian purposes, or has it always been having these many diverse functions?
Yeah, I think that, well, the jury's still out. I think that it has, multiple separable functions in the cell. And I think that its function in the clock is probably secondary to those original functions in the basic cellular processes of, regulation of transcription, regulation of splicing, and DNA damage. It's a little, it's sort of a hard question. It's a hard thing to prove. I mean, we could get it in part, I think, by looking at, for example, in Drosophila, what aspects of clock regulation are perturbed in those mutant flies. And we also, I think, can get at it a little bit with genetics in Arabidopsis, because we have multiple alleles of this gene that affect different subsets of phenotypes. Mm-hmm. So I think that's going to be powerful once we get all those phenotypes assayed.
Right. but XCT isn't a transcription factor, then, not a
It
sort of
isn't.
bona fide transcription factor.
No, no, although it's nuclear localized.
And how does this pathway interact with all the other sorts of controls? Actually, I'm presuming there are all sorts of other controls on circadian rhythms. Is the whole thing a very complex network of interactions?
Absolutely. I think that's true for any organism, you know, even cyanobacteria, that we perhaps is the simplest clock system that is generally worked on. You still have interactions of transcriptional networks and extensive post-transcriptional regulation, even in cyanobacteria. And certainly that is true in plants and animals and fungi as well. And I think kind of proteins such as XCT, I believe, and certainly the PRP19 complex, I think they're important in coordinating different aspects of nuclear processes. So coordinating events like transcription, like DNA repair, like RNA processing. And of course, all of those things are intimately connected to the circadian clock as well. So it would make sense that you would want to tie them together in some way.
Well, maybe you've already answered what I was just thinking then is, are there come on, think of reasons why this has this become a complex network of interactions, just in a sort of Rubik's cube, just through evolution, or come on, see what functions a complex system like this has. Does that make any sense?
Yeah, well, so I pointed out those. I don't think this is unique to plants at all, the complexity. But I will say that the plant circadian oscillator has significantly more components than the oscillators of other organisms like mammals or flies or Neurospora or cyanobacteria. So those have, even if you count auxiliary loops, maybe you're looking at half a dozen, maybe 10 max transcription factors. But if you look at plant oscillators, I kind of lost count, and keep more components keep being identified all the time. But dozens of transcription factors work together to make the plant circadian oscillatory network. So even, yeah, so for sure it's complicated. Plants are even more complicated, I think, than other clock systems. And I think maybe this isn't really answering exactly the question you asked, but I think that the reason that plants clocks are so complicated is that the optimization and anticipation of responses to environmental cues, I think, is even more important for plants than for animals.
Right, right. You mentioned a moment ago that you're also doing work not just under laboratory conditions, but outdoors in the field. And is this the work that you've been doing with sunflower?
Yes, so we've become interested in understanding how the circadian system influences sunflower growth and development. And kind of as you brought up earlier, you might wonder why sunflower, because it does not have the amazing molecular genetic tools that Arabidopsis and some other plants have. But what it does have is really fascinating biological regulation. And in particular, the two processes that we've become very interested in are the solar tracking of young sunflower plants. So they're of course, they're famous for following the sun. They bend from east to west during the day, and then at night they bend back from west to east in anticipation of dawn. So that's something we're really fascinated by. And then the other thing that we've become fascinated with is floral development. So it's a sunflower is an Asteraceae, or sometimes called compositae. It has a composite floral structure. So you have, in the case of domesticated sunflower, hundreds of small flowers pressed together to make the capitulum, or the head. And it turns out that the circadian clock also regulates the timing of floral development on that head. So we're quite interested in that as well.
And that flowering is regulated with what purpose in mind? That can't all these little flower things open up just at random in the course of the life of the plant?
Yeah, so you would, yeah, so they could, but they don't.
Yes.
So,
right. So why don't they? What's
Yeah.
the advantage for the plant in having synchronized flowering of all these?
Right. Yeah, so we were really struck when we started paying attention to the plants in the field. We were really struck by the fact that, so if you have a big capitulum, it can be, 12 inches across even. If you, and you can see this even in flowers you get from the florist, you'll see that the florets on the outside perimeter of the head develop first. And then over the course of about a week, the maturation goes from the outside in to the middle of the head. And it's quite striking. You probably, if you Google Fibonacci series sunflower, or you'll see that the immature head has these beautiful spiral patterns of floret arrangements on the head. And that echoes the timing of early floret development. So they develop in a spiral pattern very early on, but late in development, that spiral pattern is transformed into a ring-like pattern. So if you look at a mature head, like you buy at the florist, you'll see rings of different flower anatomy on the head. And what is happening is that in the morning of one day, dozens of florets within one ring-like region will coordinately undergo late-stage development. So they'll all release their pollen in the morning, say within a half hour's time. Across this big head, they're just incredibly coordinated and synchronized. And then the next day, those flowers will release, their stigma will emerge. So that's the female portion of the flower. And that all happens coordinately as well. And then the next older set of florets will be releasing their pollen. And so I think that it's probably, I think the reason for the coordination is to make them quite appealing to pollinators, like to say, okay, now's the time to come and get your rewards. And of course, at the same time, remove pollen and take it to another plant for pollination services.
Well, I've kept you long enough, I think. So next time I go to the florist, I will look much more carefully at my sunflowers there.
You should. They're fascinating.
Stacey, thank you very much.
All right. Thank you very much. I enjoyed it.